Good morning, grade 11 students. I hope you're doing all well. Today is Monday, March 16, and let's continue Lesson 1, Part 2, about viruses. Please make sure that your books are in front of you and to open them on page 574. The objective of this section is to compare common cold and HIV and relate them to lytic and lysogenic infection. Two examples of viruses that contain an RNA as their genetic information is the common cold and human immunodeficiency virus, or HIV. The common cold. What happens when you get a cold? First, a capsid settles on a cell in the host's nose and is brought inside the human body. After the RNA is inside a host cell, the viral protein starts making copies of its viral RNA. What happens next? These viral RNAs that are made will direct the cell to make viral capsid or protein coats that are the outer membrane of a virus. The new capsid assembles around the viral RNA, and within 8 hours, the host cell releases hundreds of new viruses to infect other cells. To recap and show how a common cold resembles a lytic infection, it starts by a virus attaching itself and penetrating into a host cell. Then it tricks the DNA and chops it up. After that, it starts replicating and making viral RNA that produces viral capsids, which later gather around the viral RNA to make thousands of new viruses that burst and travel to infect other cells. Let's move on to discuss HIV, or human immunodeficiency virus. HIV belongs to a group of RNA viruses called retroviruses. We all know that a DNA makes RNA to later make proteins which direct cell function. However, a retrovirus is whereby genetic information is copied from RNA into DNA and not the other way around. The RNA virus makes copies of the viral DNA and inserts it into the DNA of the host cell. That resembles a prophage in lysogenic infection. The viral DNA is inactive for many cell cycles, and once activated, it targets the human immunity system. This is a comparison chart between lytic and lysogenic infection. A lytic infection, after viral reproduction, the cell lyses, whereby in lysogenic infection, the viral genetic material is integrated into the host cell DNA. In lytic infection, it does not have a prophage. However, in lysogenic, it has one. In lytic infection, viral DNA or RNA replication occurs independently from the host's DNA replication. However, in lysogenic, viral DNA or RNA occurs along with the host's DNA replication. In lytic infection, it occurs over a short period of time. In lysogenic, it may take years. In lytic, it cannot follow the lysogenic infection, while lysogenic follows the lytic. Finally, this is also a comparison chart between viruses and cells. Viruses must infect living cells in order for them to grow and reproduce, while cells have their own independent sexual or asexual division. Viruses have DNA or RNA, while cells have only DNA. The viruses contain capsid, while cells have only cell membranes. End of Lesson 1, Section 20.1, Part 2. Thank you for your attention.